Hi, this is Diana. Today we are going to talk about the Mannitol Salt Agar. So first, what is mannitol? And mannitol is a sugar alcohol. Basically, it is a carbohydrate, which is a food source for the organism. And next, the salt. There is a very high concentration of salt in this agar, which is at 7.5%. It's pretty high, and it's to inhibit the growth of most bacteria. And next, the phenol red is another component in this agar. It is a pH indicator. And also, it gives this Mannitol Salt Agar a bright pink color. So the pH indicator will have a color change when it detects the acid. So when the acid is produced from the bacteria that can ferment the mannitol, it is going to drop the pH, which is going to change the color of the pH indicator, then creating a yellow zone. So which means the phenol red is going to turn to yellow. And also, it is a selective medium, which is for organisms such as the Staphylococci species. Which can live in the high concentration salt agar. And meanwhile, it is a differential medium, which is to distinguish if the bacteria are able to ferment the mannitol or not. And here I have made a flow chart to show you how to record the Mannitol Salt Agar test result. So first, you need to check if there is a bacterial growth on this agar. If there is no bacterial growth on this agar, you should record as a growth negative. And however, if there is a bacterial growth on this agar, you should record as growth positive. So the next, you need to check if there is a mannitol fermentation. So how to check the mannitol fermentation? Like we mentioned here, if the phenol red turns yellow, which means there is an acid produced by the mannitol fermentation. So here on this agar, on the right side, we can see it turns yellow, which means there is a bacteria here can ferment the mannitol. So we record as mannitol positive. However, if the agar didn't change the color, we're gonna record as a mannitol negative, which means there is no bacteria are able to ferment the mannitol. Okay, thank you for watching.